Okay, so I know we left off around here. We're about to talk about antibodies and T-cell receptors. I mentioned last time that there are the two different systems, that there's the B-cell system, that system, there's kind of like two major ways our immune system functions, or two kind of teams, and it's the B team and the T team. And the B team is the B cell system. The B cells live in our bloodstream. They target mostly foreign invaders, things that are in the bloodstream. The big one is bacteria. So I like that it's B, blood. B cells are made in the bone marrow, and they target bacteria. So all those B's you can kind of think about go together. The T cell system, the second one, the T cells, there are let me go back. The B cell we call the humoral immunity. The humors are the blood. So B, blood, humors, bone marrow, bacteria. The T cell system is our cell-mediated immunity. That these are the team that go into our cells that have either been infected by a virus or are cancer cells. And the T cells are differentiated and made in your thymus. So both in terms of recognizing any kind of cells that are not supposed to be there or are infected, they use the surface markers, and I know we talked about that last time. So we're going to just look at the B cell system, which produce antibodies. The T cells do a very similar way of identifying and remembering any invaders from before in our body. So all of our cells in our body have all of these surface markers on them. One of the particular surface markers are your species marker. So every species has a different marker. So let's say that in humans, ours are shaped like this. We have ways that our white blood cells just go up to cells, and they'll go up to red blood cells, they'll go up to other white blood cells, they'll go up to cells like mast cells, whatever cells in our body, and they go check, okay, cool, check, check, and they bind together and they make sure that they fit together like two perfect pieces, and they are like, okay, cool, we're good. Those are our cells, we can move on. It's when they go up to a cell, and if the cell is, let's say their receptor is flat, and they're like, uh-oh, this doesn't work. What they call this thing that's sticking off a foreign cell is, we call it an antigen. So even though it's a surface marker, we specifically call it an antigen, and the antigen is we're going to make an antibody that perfectly binds with that particular surface marker or antigen. So again, B cells, we're going to call them antigens, and B cells and T cells, we call them T cell receptors. So we're going to focus on the antibody antigen system. Okay, so antibodies are produced by the B cells, and the T cells produce T cell receptors. So we're going to explore what the antibodies are and how they are created. So an antibody, it will bind, and it will make a way to bind with a specific antigen. So here's the thing, is that when we have foreign invaders come into our body and they have their surface markers, let's say, again, shaped like this, that when our cells go up to them, they're like, oh wait, those don't fit like two perfect pieces of a puzzle. We don't have a receptor that binds with the antigen or the surface marker from the foreign invader. So we have to make that. And that's what the antibody does, is the antibody produces a way to bind and make a binder that fits perfect with the antigen or the surface marker of the foreign invader. That's what this whole system does. So the antibody structure, it's kind of interesting. It's shaped like a letter Y. You've got nice pictures of them on the right side of your notebook there. There's two parts of the antibody. There's the constant region, which is like this part of the Y. The constant region is the stem of the Y, or the bottom of the Y. The top part of the Y is called the variable region, and it forms the V part of the Y, the top of the Y. You can remember this because variable begins with a V, and the variable region forms the V part of the Y. So, it's a really, our body, one of the principles of living things is that living things are organized, and part of the organization of the immune system is that we have this part of antibodies already made. They're like blanks. And then we're going to add on these specific parts on the top. So here's the idea. Let's say that you need to get a new key made. And if you walk into, let's say, Home Depot, and you have the key that you need to be made, you go up and they have a wall of blanks, right? And those blanks, then what they do is they're like, the person will try and find one that looks close to your key, and they'll take that blank down, and then they stick your key in a machine, and they stick the blank in the machine, and the machine, it goes and it traces your key, and it carves that into the blank key. That's like what this is. This part of the antibody, we've got all kinds of blanks ready to go. So that the only thing we need to do is when a foreign invader comes, is we need to carve out a specific binding site or specific binding sites on the variable regions of the antibody so that they can perfectly bind. But this part is already made. Imagine that you go to, let's say, you go into Home Depot and you're like, okay, I got 20 minutes, I gotta get this key made, and then I gotta get to my next place. And you walked in, and the person's like, oh, okay, you need a key. Okay, let me go in the back, let me heat up the metal. I need to pour that metal into a mold shape, and then I'll be back in about an hour, and then I'll be able to carve out your key. That would be ridiculous, right? If everybody had to wait for them to pour those blanks and make those pieces of the keys. So we don't have to do that. It's just like walking into the Home Depot. You just pick a blank, and then you add on the carved pieces that are specific to the foreign invader's surface marker, to their antigen. So that saves us a lot of time. We can see that within our antibodies and our body, here's 3 different sicknesses that are caused by foreign invaders. I want you to notice that the bottom part of each of the 3 different and very specific antibodies are the same, right? Same, and same. And what will be different for every different foreign invader is the variable region will be specific to their surface marker or antigen. Triclosan is a common antibiotic used in antiperspirants, toothpaste, antibiotic soaps, and lotions. And you can check this. They've been phasing this out for a while. So I'm always a big proponent of read your labels. This is one you want to look out for. Which of the following is most likely, which of the following is the most likely prediction about this antibiotic? So it is as effective now as when it first came out. Does that match anything we talked about with evolution? No, probably not, right? Because we know that bacteria, they have high mutation rates. There are no microbes left that are resistant to this antibiotic given its widespread use. Does it go in the other? No, that seems like it goes in the opposite direction of what happens with microbes. There might be microbes left that are resistant to this antibiotic in areas where these products have not been distributed. So there's natural selection, right? That where the microbes live and this is used, microbes are going to probably not be favored, microbes have evolved to become resistant, and only those that do not have resistance are favored and live on, but they probably are all resistant at this point. So in areas where this hasn't been present, it will still persist. The microbes will persist. And everyone who's exposed to this antibiotic and doesn't use this product will die. Really bad. Okay, so the C is correct. All right, so what about the issue of our self cells? Sometimes our self cells get attacked too. On the surface marker of our self cells, or what the surface marker of our self cells that identify us as individuals, not just as a species, but we're talking about the difference between you and I. We call them MHCs, or major histocompatibility complexes. So these are unique to us as individuals. I have a different one than you do. You have a different one than the person next to you. So not only do we have species markers that we all will have as the same, but we each have individual MHCs as well. They are unique to each person. Okay, so here's the thing. Let's talk a little bit about what if you're starting to have kidney failure. And because I like you so much, I'm like, I will give you my kidney. I have two. I can live with one. I'll give you one. It doesn't work as simple as that. Even though you need a kidney and I'm willing to give you one, we have to make sure that our MHCs match close enough. And even though I want to give you a kidney, and even though you need a kidney, let's say we just haphazardly, we're like, let's stick her kidney in me, and we'll see how it goes, without testing it out. And so what can happen is that your body, even though your body critically needs that kidney, and you have this new functioning kidney in you, if those MHCs are not closely matched enough, they don't look close enough, that your immune system can go, no, this doesn't belong here. This is a foreign invader, and we're going to attack and destroy it. So the idea of organ transplants, there is a lot of testing that goes on to ensure that your MHCs and my MHCs are close enough that your body's immune system can be kind of fooled into thinking that my organ is one of your own. Kidneys are relatively easy. I'll say relatively easy. Usually you can find someone for a kidney donor, sometimes in close family and sometimes not. It's kind of odd that you'd think that someone like a brother, sister, mother, father, aunt, uncle, cousin, and sometimes it doesn't work out. Sometimes it's a total stranger that matches better to you, and we don't really understand why. Blood donation, we know that there's different categories. We have blood types A, B, and O, and those have to stick together, but also the Rh factor has to stick together, that we have plus and minus, A plus, A minus, O plus, O minus, etc. So those are a little bit easier because the rules are quite flexible. You just have to stick within your blood type and Rh factor. And things that are much harder, hearts, lungs, those are much harder. Lots of research opportunities here in this field. All right, so job 2. Once we have identified something, our surface markers do not fit with the surface marker of something else or the antigen, we have to get that. We have to acknowledge that something is there, and then we have to make the system to start to attack it. So part 2, what happens is on your B cells, and again, humoral immunity. See, this is where I put all the stuff that I should have put far earlier. The B cell system, your humoral immunity, it attacks in the bloodstream. Mostly bacteria, can really be any microbe. The B cells are going to go, okay, this, we don't match, they're just gonna grab onto it. So they're gonna grab on and hold on to it. Even though they don't bind perfectly, they do, certain B cells have a system to be able to grab onto the antigen of a foreign invader. Once this binds, this particular B cell binds onto the antigen of the microbe, this starts to clone itself really, really quickly. And it makes lots and lots and lots of clones. The clones, in this meantime, the antibody is going to be formed to match perfectly with the foreign invader. And so now, once this first one is made and it makes a carving of the variable region, then it clones itself and makes lots more. And then the clones become memory cells, so that now we have B cells that have surface markers that match up with this specific foreign invader, and we keep that as a memory on our cells. So that if this particular bacteria comes back again, then we can immediately, instead of taking the time to, carve this out, just like a key carved out at the Home Depot, we have this already to bind perfectly onto there. Next, we also make plasma cells. So, I like the illustration. Let me talk through the illustration. Okay. And then I'm going to walk through this when we get to the picture. And talk about the role of these macrophages. Okay, so I like the picture because it goes through what is actually happening. So first thing is that you have the first cell that encounters the foreign invader. And that first cell is going to take a little bit of time. It's going to carve out the antibody that binds perfectly with that foreign invader's antigen or its own surface marker. Now, this one that has those perfectly formed antibodies is going to clone itself. And then the clones clone themselves. And when we get to about the third or fourth level, what happens is that the clones, they are going to start producing more of the same cells, but then they're also going to produce plasma cells. And this is pretty wild, that they can produce two different cells from one kind of cell. And this plasma cell, it starts to just produce mass amounts of antibodies by themselves. So that now, instead of a whole cell has to seek out, and remember that they are bacteria, and the bacteria in our body, they are replicating quickly as well. So every 20 minutes, so remember that this has to make a new cell. It's going to take longer for us to make one of these cells than it is for them, the bacteria, which are much smaller, to make more of themselves, to clone themselves. So the bacteria are getting a little bit ahead at this point, that they're making more and more and more bacteria. So they're ticking up, and we're slowly making our antibody and then cloning. And so our army, essentially, we are starting to make this whole team that is going to work on them. But as we're building our team, we're slowly building our team, they're reproducing at a much quicker and higher rate. Also remember, though, that at this time we are very likely having a fever response. And so that our second line of defense in our immune system is that when we have a foreign invader, we are going to start to have those endogenous pyrogens are going to tell the brain, hey, let's start upping the temperature a little bit, and that fever response is going to help our immune system. It's going to help our immune system do this a little bit faster, and it's going to make them slow down a little bit. So initially it's like this is going fast, and then the fever catches up, and then we're going to start seeing this happen. And hopefully then this is going to overcome that. So kind of interesting timing wise, it takes a little bit of time. So the plasma cells, as a product of a few generations later of our original B cell that made the original antibody, the plasma cells, they're just going to pump out tons of these antibodies that now these little antibodies on their own, the little Ys on their own, they can just float around in the blood. And when they, by themselves, find a bacteria, they go onto it. And when they grab onto the bacteria's antigen surface marker, then that signals to a whole different team of white blood cells, the macrophages. And it says anytime a macrophage, which is like little, like a Pac-Man, they go in the bloodstream and they go, oh wait, this thing here, one of these has a Y on it. They're like, I eat anything with a Y. I eat anything that has an antibody on it. And then they start to gobble up. So that you have these original B cells that are just trying to pump out, to make new B cells and pump out the antibodies. So that's one part of the immune system. The other part are the macrophages, which are just floating around to look for anything that these are bound to. So two different kinds of cells here that are working for the same goal. And just another picture of the same thing. But here you can see these are different antibodies. I like this picture because it shows that in this case, this cell, these are old invasions by other bacterial cells. And so you've got some memories of old infections, whatever, this one in green, this one in blue, purple, black, all of these are different former foreign invaders. And now we have a new foreign invader. So these yellow bacteria are new. And so we create a brand new antibody, the red. And then you can see of these five different kinds of B cells, the only one that takes action to replicate is the red one because it is now specific to this foreign invader. So we've got lots of memories of now different kinds of infections in our body. And then this one alone will replicate and then replicate a few generations later to produce those plasma cells and just pump out lots and lots of antibodies so that the macrophages can notice anything that has one of those red antibodies on there and eat those cells up. Okay, cell-mediated immunity, just to mention, the T cell system. The T cell system does a little bit different things. It is inside, and what I mean by cell-mediated immunity is that this happens inside the cells. So the B cell system, the humoral immunity of our adaptive immune response. Remember, we're talking about the adaptive immune response has two parts. It's got the B cell system, the humoral immunity, and it has the T cell system, the cell-mediated immunity. So it's got two teams for different issues. Our second team, cell-mediated, this happens to cells that are either infected with a virus, they're taken over by a virus, or cells that grow out of control in our body are cancer cells. The T cell has a bunch of different kinds of cells, just like we talked about before. You've got the B cell, and then you've got the macrophages. The T cells have what are called cytotoxic T cells. They're the ones that do the binding. So this cell, this cytotoxic T cell, going around the body, it notices that you have a cell that's growing out of control. And it's going to, you can see, this is a great image, that you can see the cytotoxic T cell has bound with the cancer cell, and it's going to start gobbling that up. It's using enzymes like lysozymes that were found in our mucus to start to dissolve these cells. You have helper T cells. The helper T cells, they're just there to give a boost of energy, of anything that the cytotoxic T cell needs. They help them. I like that they're called the helper T. They help the cytotoxic T cells to gobble up those cancer cells or the virus infected cells. Then you have the suppressor T cells. They're like a project manager. The suppressor T cell project manager is going to watch. They're going to go, hey, okay, I guess this cancer group of cells, they're watching the cytotoxic T cells, they're watching the helper cells work together to get rid of those cancer cells, or you've got a bunch of cells that are infected with viruses, and the suppressor T cell, like the project manager, is just going to kind of watch. And when all of the cells are pretty much eaten up, dissolved, then the suppressor T cell tells the helper T and the cytotoxic T, we're good. Everybody relax. We got this under control. It ensures that we don't waste a lot of extra energy of the helper T cells boosting the cytotoxic T cells, because we've got other things to do in the body. The body is very busy, so we don't want to waste a lot of energy. So they're going to just say, everybody, okay, good job. And then you also have memory T cells. And the memory T cells are going to have receptors on them to remember any cancer cells that have formed before in our body, any virus infected cells. So both B system, the humoral immunity, and the T system, the cell-mediated immunity of the teams of our adaptive immune response, they both have memories of what has gotten us before. So just lots of great pictures of immune cells in action. I just think this is so cool to see, like, not just an illustration, but you see what they really look like. All right, so again, remember, you've got your humoral immune response. This happens in the blood. It's the B cell system. They mainly target bacteria and other microbes in the blood. The B cell encounters a foreign invader. It's going to quickly take a generic antibody and it's going to make it specific to binding onto exactly that foreign invader's antigen surface markers. That B cell is going to quickly replicate itself to make cells that have that antigen on there, the new antibody on there, and then also makes these plasma cells to just start pumping out tons of antibodies. Any cells that one of the memory cells binds to, or an antibody binds to, macrophages come and they eat up those cells that are tagged. Inside of our cells, we have the T cell system, the cell-mediated immunity. The cell-mediated immunity will detect virus-infected cells as well as cancer cells. So you have your cytotoxic T cells. They are going to immediately start to bind to this cell has virus particles inside of it. Could be cancer cells. It's going to form T cell receptors that are going to perfectly bind with the antigen surface marker of your foreign invader, foreign invaded cells, and also any cancer cells. Helper T cells are going to give a nice boost to keep the cytotoxic T cells working hard. You have suppressor, the suppressor's hanging out somewhere, watching the system, so that when this is under control, these virus-infected cells or cancer cells are eaten up and dissolved. Then they say, okay, we're good. And then you have your memory cells for the future. Okay, so memory. As we keep saying, B and T systems both have memories of past invaders. So let's say you got the flu really early in the flu season. I was talking with a geneticist last week. I was at a conference, and he said that flu this year, so every year, what they do with the flu is about now for next year. For next year's flu season, what scientists are doing is they are analyzing the genes of the flu that's going around right now. And they're sequencing it. And based on the sequence of the flu this year and previous flus, and they look at how the flu has mutated over the years, they predict what the flu next year is going to do. They make a prediction. Do you think they're always right in predicting how the flu next in the 2026 fall? Do you think they always get it right, how it's going to mutate? Not always. And sometimes they're way off, and we have flu seasons that get really bad because the vaccine that these scientists are producing, it doesn't match well enough. What they believed happened this year was that it was close, but not close enough. So if you got the flu vaccine and you still got like the flu, like I know I got, my whole house, we got it. Could have been a cold, could have been the flu, but a lot of people got either a mild version of the flu, which is what happened in my house. We all got a mild flu. Usually me and my older son get high fevers when we get the flu, or we get some like really bad sickness, and we didn't get fevers at all. We didn't even get a low-grade fever. But we had like all of the congestion and feeling run down and exhaustion. And people who didn't get the vaccine, they got it really badly. So he was saying he believes that they got most of the genes of the vaccine, but not all of them. And that's why some people got like a more mild version of the flu. So kind of interesting. So right now, just topically, one of the things that DOGE, our government system right now for cutting out money and waste, is that they cut the funding to the people who are starting to research the flu for next year. So we'll see if they put that money back. I really hope they put it back. Because people will die from the flu. It is something that happens. But maybe next week, we'll see they put that money back. Okay, so ever wake up in the morning and you're just like, oh, I feel a little off. And you just feel like, I know I'm getting sick. You have that feeling, like I feel kind of tired, your throat's a little bit scratchy, you feel a little run down, you feel like fuzzy, and go throughout your day and you're just thinking like, I'm sick, I'm getting sick, I know it. And then you go throughout your day and you go to sleep, you don't come home and you're just tired, and go to sleep early. You wake up the next day and you feel good. What probably happened there was that you had a return of an invader that has already given you a pretty bad sickness. That our memory cells, that you had a second exposure to something you had in the past, and you have this like little mini, really quick immune response that might take just 12 hours for those memory cells to just start replicating, because they already have the specific antibody to match the antigen, and you don't have to go through that whole system of creating that team that's going to start gobbling them up. So before, when we talked about like the bacteria, for example, will start to replicate, and then we're slowly taking up and getting closer and closer to them. What can happen is the bacteria, we're on pace with them. And so we're basically, our system, because we already have those antibodies made, they can just kind of, in time, as the bacteria replicate, they're gobbling everybody who's replicating up. And so you have like this little mini war that happens in a day, and your body feels a little bit run down, but you don't get the, like let's say you got the flu this year, and you don't have those like week or 2 weeks where you just feel awful. Because in those 2 weeks, the bacteria or the flu virus, the flu particles, whatever's infecting you, it's up here and you're like trying to catch up to it, right? And then overcome it. So this is like a full-blown war upon first exposure that happens in your body. And then you have a couple weeks where you feel weak. And your body is basically like making up for all of the white blood cells that have given up their lives to destroy any foreign invaders. You have to remake those. So your body is still recovering. And then in maybe like 3 or 4 weeks, you feel back to normal once from start to end of when you get sick. But those just kind of little days where you just feel a little off, probably have a return visitor that is getting taken care of in maybe 12 hours. So it's pretty cool that our body does this. All right, so sometimes our immune system needs more help than normal, and we'll use things like antibiotics or vaccines. What these do is they help our immune system. They help our immune system to finish off the job. So now, like if we're thinking about, okay, here's whatever's invading our body and it's going fast, 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 and then we're trying to get there, we're gonna add a third thing in. And this third thing in is going to help us get there faster. So that's all that they do. The immune system, they don't actually solve the issue, they just give a boost to the immune system. All right, so antibiotics. Antibiotics, they can treat a wide variety of things. Antibiotics do not only treat bacteria, they can treat fungal infections, protist infections. They don't treat viral infections, though. What antibiotics do is that they are individually specific to the invader group. So some of them might start to digest part of the cell wall. Some of them might start to attack the genome of the bacteria or the protist or the fungus. So they'll do different attack mechanisms depending on the group that they are trying to attack. I was just reading an article about AI. I thought this was really fascinating, the way AI is helping a lot of fields. I'm trying to learn a lot more about it. So there are scientists that study soil around the world. They collect soil from all over the world and they test that soil to see if there are natural antibiotic properties in soil in different areas of the world. And the way that they manually, they do something manually in the lab to do all these tests. And some labs have started using AI to test the soil samples on the microbes for antibiotic properties. And it's like something ridiculous, like 1 trillion times faster to use AI to do it than for us to do it manually. So I know sometimes we think like with AI start coming out, everybody's, like, is it good? Is it bad? What is it going to be bad? But there are, and especially in science, we're going to find as we move forward in our futures, especially your futures, that AI is going to help us to do our jobs even better. So interesting things happening. Okay, so antibiotics are good agents of natural selection, that they can help our immune system, give it that boost. The antibiotics select for any bacteria that do not have resistance to them, and they knock them out. Opposite way, for thinking about the bacteria, like when we were talking about evolution, that any bacteria in the presence of an antibiotic, any bacteria that have antibiotic resistance to that antibiotic, they are selected for. So it kind of works both ways. All right, so a little bit about vaccines. What are vaccines? Vaccines are ways to quickly excite our immune system without having an actual infection. They get our immune system to make antibodies or T cell receptors against something. Could be bacteria, could be a virus. All right, so what is a vaccine made of? Vaccine can be made of live virus particles. I mean live, like I'll say active. I don't like to call viruses live, but active virus particles. They could be dead microbes that you put bacteria that are dead in there, but they have the surface markers still. Or it could be just some of the antigens of the pathogen. Could be antigens that are on the surface of a virus or antigens that were on the surface of a bacteria. Depends on the vaccine, what you're going to get in there. And so what happens is without you getting an infection from the virus or the bacteria or whatever the vaccine is intended to help you with, you get just exposure to their antigens, and then your body makes antibodies. But here's the thing, that you're like, well, wait, if it's making antibodies, aren't you fighting some kind of like infection? No, because the infectious agent is inactive or dead. So here you get to just like casually, your immune system casually gets to make antibodies without suffering from the infection. That's great, right? That's the whole point of vaccines, is so that you can have this like little mini immune response without having a full-blown immune response to the agent of infection. And so you get the benefits of not having an infection, but getting the protection. That's great. The controversy about a lot of vaccines is that people read, there's one particular study by Andrew Wakefield, who he had surveyed 12 kids who had autism, and after getting the MMR vaccine, which we're hearing a lot more about today because that's the measles, mumps, rubella vaccine, that he made connections to them having symptoms of autism that only came after they got the vaccine. There were only, I can't remember if it was 8 or 12, I can't remember which one, but it was a really small sample. It was super manipulated, the study, and there are mass studies now from around the world that disprove, that do not support that the MMR vaccine causes autism, or that any vaccine causes autism. So there's a big history why people glom onto that as saying, like, I'm not getting my kid vaccinated because vaccines cause autism. And with the ability to produce so much misinformation out there on the internet, right? I could write a whole blog and make up a whole study. I could make up all these charts and graphs that look really good to support my point, and none of it be true. And you might come upon it, or some, influencer decides they're going to make a little short video that highlights what I put, and now it's out there and millions of people are like, that sounds really good. She's very convincing. We've got to be careful about what we see out there. So, no infection, but you get the memory cells made. That's what the vaccine does. Cancer cells. Cancer cells, our cells just uncontrollably grow. Some of these are what we call benign, that we can have cancer cells. You could have a cell that has a mutation to its checkpoint function, and the checkpoints just make sure that everything goes okay in your cell. You could have a mutation to one of the genes that prevents your cell from replicating over and over and over again. And you have a mutation for one of those genes that makes your cell replicate and then go do its job for a while. And it does its job until it's run out of mechanisms to do it. It needs to be destroyed, and it replicates. So sometimes those genes that prevent our cells from replicating over and over again, they can just by chance have a mutation to them, and it causes a cell in your eye to just start to replicate without being malignant, meaning that it's not going to cause you to have some kind of like awful disease in your eye, but it is replicating a lot. And so then your T cells will gobble that up. When we talk about cancer, we're often talking about the ones that cause disease or cause us death. And cancer can be caused by so many things that there are a lot of environmental factors that can mutate our cells to cause us cancer. A lot of times though, it's in our genes that we have the genetics. And then sometimes it could be this isolated, could be that isolated, but a lot of times it's that you have some kind of mutation that gives you the predisposition to having a certain kind of cancer when you are in a certain environment. So like for example, my father had melanoma all over his face, one on his back. So I likely have the genes for melanoma. Now, if I always put on sunscreen, wear a hat, wear sunglasses, protect my skin, it limits my environmental factors for the genetics that I have. And so I can control my environment to limit whether my genes get activated or not. And so like in my family, we always wear sun shirts and sun pants, and we cover up, wear hats and lots of sunscreen because I know that we have those genetics. Sometimes viruses will cause cancers. Whatever they are, they sabotage the mechanism for replication, cause those cells to replicate out of control. All right, so immune system, like I keep saying, our T cell system recognizes most cancer cells. Again, cancer cells are just cells that grow out of control. But the malignant ones, the ones that are more dangerous, they have tricks. And so some of them, they might just lack certain proteins that are found in our body. And so then our cells are like, well, the T cells they go, well, I don't know what's going on with that cell. It doesn't have the right proteins. I'll just ignore it. And it tricks our cells into ignoring it. Or they don't have the right surface markers. Maybe you're looking for some kind of different mutated form of our individual surface marker or species marker or some other surface marker. And in that case, maybe that surface marker is just not present in a cancer cell. So then your T cells are like, there's no thing that I can even try to bind to, and they just go, okay, fine. And for whatever reason, they just ignore it. So it's kind of like our O blood cells, that our O blood cells, they do not have a glycoprotein attachment for notation of what blood type cell they are. They don't have that surface marker. Because they don't have that surface marker, O blood cells are really valuable to us in terms of blood transfusions. Because they don't have a surface marker, they're kind of like a blank blood cell. They're like a filler that our body won't attack them because they don't have a surface marker. And so like your surface marker, the blood cells, the B cells go up to it and they're like, no surface marker, so they ignore it. And in that case, it's a good thing because O blood cell can be a good filler. In the case of a cancer cell, no surface marker, that cancer cell will replicate and replicate and replicate and then cause certain symptoms to happen in our body, maybe certain poisons, they produce some kind of toxin. And so there's a lot of different ways that as they grow out of control without having a surface marker, we don't recognize what's happening. So really weird. There's tricks that those cancer cells are tricky. And inside of our cells, specifically your natural killer cells and your cytotoxic T cells that usually are watching for these different surface markers, the antigens, they don't see any. And they just go, okay, I don't know. I'm gonna let it go. So there are lots. I mean, there's so many different forms of cancer. There is so much research that's going on. There's so much potential for researchers, medical professionals, genomists, lots of people out there who can figure out the trick of each different kind of cancer and tailor medicine, or even it could be something that they create a virus that attacks those cells. So there's lots of different ways to treat, and lots of research is going on for cancer. Lots of cool stuff happening, lots of potential out there. Okay, so vaccinations also can prevent some cancers, like hepatitis, liver, mouth cancer, throat cancer, penis cancer, some leukemias. We know cervical cancer, that it's recommended that kids at about the age of 11 to 13 get Gardasil. So we do have vaccines that can prevent some cancers. So that's, again, lots of great research happening out there. Okay, so some of the vaccines, like to prevent hepatitis B, which will trigger some cases of liver cancer. And as I mentioned, the Gardasil, HPV. Somebody discovered a vaccine that can combat certain strains of HIV, but they are the very less common strains. So we're getting closer in that way too. Okay, what happens when your immune system just totally malfunctions? We get allergies. This is where your immune system basically overreacts to something that is not harmful. So all of these things, could be pollen, could be something not necessarily devastating to you, like every year. I know now mold, has been raining a lot. I know that the mold is growing out of control with all of the moisture, and my nose is just running. But I know I have a mold allergy, that usually in the fall and in the spring, when the mold spores are just popping, I'm going to get a lot of congestion. Could be pollen, that maybe in another month when things start to grow, you might get that overwhelming kind of like light cold flu symptoms. Bee or wasp venoms. My office mate, her father-in-law, he is a beekeeper, and he got stung one day and he went into cardiac arrest. He had a heart attack because of the sting, and he was like, it's crazy, he was dead for 6 minutes, and now he's like totally alive and healthy. But he has to stop doing beekeeping because sometimes beekeepers, with their genetics, they after a certain amount of stings, they have this overblown immune response. Some people are allergic to foods, right? Anybody have food allergies? So like in schools now, you can't bring anything tree nut or peanut related into schools, because some kids can go into anaphylactic shock where their throat swells up so much. It has an inflammatory response so strong that it closes up the trachea. So these are called, instead of antigens, they're called allergens. Or I guess the antigens are called allergens, I should say. So basically in our system, what happens is that your mast cells get activated. And when your mast cells get activated, they send out histamine. And what histamine is supposed to do is it's supposed to activate the inflammatory response so that it opens up your blood vessels a little more and it adds water to the area of infection so that you have more space for your white blood cells to do their job. So when these kind of benign things activate the B cells and then activate the mast cells and then activate the histamine, that you get this swelling. And we, like I mentioned last time, that we're hearing a lot more about the dangers of inflammation in our lives, and we're not sure, we know that's not good. Especially if you get like seasonal allergies, inflammation, one of the things that it does is that the mold spores or the pollen that come into your lungs, it causes your mast cells to release histamine and causes more water within your lungs. And that water causes mucus to form, and the mucus and the water then cause the harder breathing and the coughing and the sneezing and the runny nose and all of that. But that can also happen in our cells as well for different reasons. And we want to limit this activation of our immune response. We don't want that. We want our immune response to focus on what it does, foreign invaders. We don't want it to go overactive over things, because what if in the spring, so now like I've got this over activation of my immune response right now, what if I get what if I get a foreign invader who's really, really bad? My immune system is already taxed. I want my immune system to focus on the bad invader. Now it's got to split its time over this ridiculous thing that's not going to kill me, and now it has less to give to the actual attacker. They often call this spring and fall, well, mostly I guess the spring, when the pollen starts being released a lot, like in the olden times when hay was growing, when you had the grass that makes hay, that the pollen from the hay would cause people to have this overactivation of the mast cells releasing histamine, and you'd get all of those symptoms. Some food allergies can cause just like cramping throughout your digestive system, too much production of water and mucus, and your immune system can lead to cramping and diarrhea. And then, as I mentioned, some people, the activation of your immune response can be so strong that you get inflammation in your trachea, in your esophagus, and it causes a swelling and you can't breathe. So, something like pollen gets into your cells. Your plasma cells are going to replicate. They're going to contact the mast cells. The mast cells are going to release all this histamine within, and then that histamine is going to cause more water flow to that area. Mucus is going to build up, and then you're going to have flu-like symptoms. All right, and then autoimmune response. Autoimmune diseases, they attack your immune system, they attack your own cells for no good reason. So here's the weird thing. You've got the right MHCs. They're your own cells. But for some reason, your immune system attacks your own cells anyway. So for example, one kind of anemia, this kind of autoimmune anemia is caused by your white blood cells attacking your red blood cells. Your red blood cells carry oxygen. Anemia is when you have less red blood cells, which means you have less oxygen, which means you have less of the ability of your cells to break down our food efficiently with oxygen. We cannot effectively do cellular respiration. Why? I don't know. I was reading an article that said that your microbiome, what foods you eat, what you're exposed to, who you live with, all of those things, and we have talked about microbiome. can have a huge influence on helping with some of these autoimmune disorders. That a lot of people, they change their diet and you think that it's an actual food, but they're really thinking it's not just the health of the food, it's all the microbes, the healthy microbes that live on that food. And that some people who have eliminated certain foods, like maybe food dyes, like anything sugar related, anything with food dye, really limits some autoimmune diseases. This other article I read said that people were going on, they had digestive system disorders, and nothing could cure it, and they went on a purely vegan diet, and they ate just anything that was plant-based and fresh, not frozen, not packaged, everything was fresh made food, and all symptoms gone. And they think it, again, probably something to do with the nutrients in the food, but they really think it's the bacteria, the good bacteria that grow on the plant-based foods. All right, type 1 diabetes. Type 1 diabetes. This is something where in the youth, a person's pancreas gets attacked. The islet cells of the pancreas that produce insulin. Insulin is super important because insulin gets released by our pancreas and our blood, and insulin helps take the nutrition particles in our blood and allows them to get into our cells. They're like an escort. They bring the nutrients and energy sources into our cells. If you don't have enough insulin or no insulin, you can't get the nutrient sources out of your blood. They just stay in your blood and your cells starve. One thing to mention about type 1 diabetes is that they're not exactly sure what causes it. They have some hypotheses that there's a genetic factor. Perhaps there's in connection with in your early stages of development when you're younger, you have an environmental trigger of your genetics. They're not sure what the environmental trigger is. Is it a virus? Is it hormonal? They don't know. So something to be figured out. There's lots of other autoimmune disorders. It seems like these autoimmune disorders are ticking up right now in our world. Why? I don't know. There's a lot of pollution. There's a lot of chemicals in our food. We eat a lot of processed food. Food dyes. There's so many things in our environment that could be causing these autoimmune disorders, and likely it's a combination of all, and maybe with combination of genetic factors. Arthritis, MS is another one. I know a lot more people. Like when I was younger, I didn't hear about MS very often. Now I know a lot more people are getting MS. It's really weird. Lupus. Lupus is one that I think when doctors, PAs, when they encounter someone who has just really weird symptoms, and the symptoms change, and some of them persist and some of them change, that they just say, let's call that lupus, because it's kind of a mystery. Most autoimmune diseases have no cures. Replacement therapy, replacing the insulin that's missing from the body by doing daily either injections or having an insulin pump, is very helpful for some diseases. Here's a tough one. Immune system suppressing drugs. So if your immune system is overreacting and then you get some kind of drug, medication that's going to suppress your immune system, what happens if something bad comes along? Your immune system is like, ugh. Immune system can't handle it, right? So that's not really a good idea either. Should you live in a bubble? Can't do that, right? Should you just stay at home, go through COVID again, where you just like isolate all the time? That's tough, yeah. And then the exotic flu viruses. How about them egg prices? That's all we're talking about these days, I feel like, right? Every day we hear about the egg prices. Where are the eggs? So the bird flu, it's an exotic flu virus. A lot of bird farms are being killed because they're being infected. The birds are being infected with bird flu, causing egg prices to go up, causing a lack of having enough eggs. Supply has gone down. So sometimes we have species-specific viruses that jump over and infect other species, and that's not normal. And we're definitely seeing a lot more and more and more these days than we ever have before. So if you get infected with the bird flu, for example, you're not a bird, so you don't have any memory of anything bird-like. So that's why these exotic diseases, they can be very dangerous. That's why COVID was very dangerous. What it is that they believe it came from bats. So a little bit about the avian flu. So here's the bird flu. Bird flu is called H5N11918. It killed more than 50 million people. Not recently, but over time. The last time it was really virulent was back in August 2012. So here's a weird thing, is that with these zoonotic diseases, these exotic viruses, that in 2012, I was teaching here, that people from your age group, that like 10-year span of being from like late teen to early 20s, were more susceptible to harsher sickness that year from the bird flu. That's really weird, right? Because you should have prime immune system, and they don't know why. One of the professors here, he was in the hospital for a month. Today, it's really attacking the birds, it's sticking to the birds, it's less efficient, but if it keeps spreading through birds, it has the opportunity to mutate. And that was one of the big things with COVID, is that when we had this stupid politicization of wearing a mask and not wearing a mask, and it's my right to choose not to wear a mask, is that the spreading of COVID from person to person to person to person to person allowed COVID to have many mutations. And the less people that COVID would have contacted or spread between, the less mutations it would have had the opportunity to make. So lots of studying going on of all of these zoonotic diseases. We'll see what happens with the bird flu, right? See what happens with our eggs and the egg prices. All right, and then swine flu, which is the pig flu. So this is my pitch for getting the annual flu vaccine, if we have one next year. Let's hope we have one. Is that there are some genes of the swine flu in our annual flu vaccine. So not only do you get protection from the human flu, if the swine flu blows up again, like it did 2009, 2010, then you get a little bit of an advantage there. I'm a vaccine proponent. I think it's good to give your immune system little exercises to get those memory cells. without having to get the full-blown infection. And then HIV/AIDS. This is believed to have come from another primate. It attacks the T helper cells. So the helper cells, remember, they are trying to give a boost to the cytotoxic T cells, and the cytotoxic T cells are gobbling up the cells that are infected. It gives more opportunity for the virus particles to infect more cells. All right, this is a good question. Would you expect someone with AIDS to have a fever in response to an infection? Okay, so if you're like, I don't even know where to begin with this, think about what level of immune response AIDS, a virus, would need. Think about the levels under that should have been activated first. Okay, so are we on yes or no? Let's start there. Are we on yes or no? You're on yes. The level that HIV/AIDS is affecting is the adaptive immune response. So that's affected, that's going to be suppressed. Does it suppress the innate immune response, level 2? No, it doesn't affect that. So should your barriers be good? Yes, right? Your first defense level, your external, your non-specific external barriers should be good, even if you have HIV/AIDS. Should your innate immune response, your internal non-specific barriers be good? Yep, they should. So we're on yes. You should be able to get a fever in response to an infection. All right. Is it yes because the non-specific defenses should be relatively normal? Yeah, that sounds good, but let's just read. Yes, because the specific immune response should be relatively normal? No, that's what's infected, what is affected is your specific immune response. So A is the correct answer. That was a thinker, right? Yeah. Okay, let's just finish up these last bits, sorry. I thought I was gonna get to nutrition. Immune system, big one. This continues to be a problem. We have seen in the last few weeks, aid has been cut off to a lot of resources for people that were given help by USAID. Is that going to affect HIV/AIDS to rise in the world? Looks like it might. So we could see these numbers just as an effect of political changes right now to go up. Just to mention, the HIV/AIDS virus is a super complicated virus. Viruses, they only have two parts to them to be called a virus particle. They are not a cell. They only have a genetic material and a protein coat. Some viruses, which are very effective, only have those two simple parts. But HIV, look at this. This is a beast. One, it has a lot of surface markers. These surface markers, our cells are like, we like those. They allow, because of the surface markers and this lipid coat, our cells allow these virus particles to merge with them, and our cells allow the virus particles to dump their genetic material inside our cells. They're tricky. That's real tricky. They have a lipid coat, they have a protein coat, they have another protein coat, and then they have their genetic material. They got a lot of protection for their genetic material too. Yeah, so they're tricky. Okay, and here you can see, this is one of our cells. These are HIV particles dumping into our cells. Look at how many and how easily. Yeah. Pretty wild. Okay, so no